Hello student. Once again welcome to Anu's classes. Today in my video I am going to discuss one of the important chapter of your biology, that is chapter number 2, reproduction in flowering plant. So in this chapter we are going to discuss about the structure of flower, male reproductive structure and formation of male gametes, female reproductive structure and formation of female gamete, pollination, fertilization, zygote formation and embryogenesis, and last part is the formation of seed and fruit. So let's start the video. So in the first page, we are going to discuss about the structure of a flower. So this is the structure of a flower, and as you know, flower has three holes. These holes are calyx, corolla, endoecium and gynoecium. Endoeciums are known as stamen also, and they are the male reproductive organ, whereas gynoeciums are known as pistil, and it is composed of ovary, style and stigma, and this is the diagram of a flower, and it has total 4 number of holes. Next, structure of pistil or gynoecium. So this is the pistil. Pistil is the means female reproductive structure. This is the structure of pistil, and it has stigma. Stigma means sticky tip is called, which trap the pollens, means pollens are come and stick here. Next, style. Style means cylinder tube-like structure that transport the pollen from the stigma to the ovary. Last, third one is ovary. Ovary means which contain the ovule, and ovary developed into fruit later. After fertilization means ovary converted into fruit, and ovule, it contains egg cell which develop into seed when fertilized. So this is the structure of gynoecium or female reproductive organ. Next, pre-fertilization. In this chapter, we are going to discuss about the fertilization. So, before fertilization, we have to discuss about the pre-fertilization structures and events. Several structural and hormonal changes lead to formation and development of floral primordia. Inflorescence from that bears floral buds and then flower. In flower, male means androecium and female means gynoecium, differentiated and developed, in which male and female gametes are produced. Next come for the first type, means structure of stem, means male reproductive organ. So male reproductive organ is the stem or endoecium. So this is the structure of stem. Stamen has two part. First one is called anther, second part is called filament. So this is the structure of a stamen. Uppermost part is called anther, and the stalk-like structure is called filament. Then within the anther, pollen grains are formed, and from the pollen grain, the male sac cells are produced. So this is the structure of the anther. Next, stamen, microsporangium and pollen grain. So stamen consists of a long and cylinder stalk called a filament, and generally bilobed anther, means stamen has a bilobe structure, and each lobe contains two theca. That's why anther is known as diathecous. So you should remember why anther is called diathecous, because it has two theca, means two chamber-like structure. The anther is four-sided structure consisting of four microsporangia, two in each lobe. The microsporangia are located at the corner, two in each theca, and they further develop from pollen sac which contain the pollen grain. Now I am going to show you the real diagram. So this is the real diagram of a androecium or a stamen. So this is, look, this is the structure of the anther, this is the filament, and this is the anther. Anther is, I have told you now, it is diathecous. Here there are how many theca? Four. Two chamber-like structure. So this, look, here we have this, it contains two lobe structure. That's why it is called theca, but means diathecous, and two lobe structure. It means, look, this is the anther. So this is one part of the anther, this is one theca, and this is one theca. In another part, this is one theca, this is one theca. It means total four number of theca are present, and in each theca one microsporangia are present. Look, this is the, in four theca, four micros. When you take the transverse section, you will, the anther will be shown like that. Next, go for the next part, microsporangium, structure of microsporangium. So within this anther, here the microsporangiums are, these are the microsporangium. Now we are going to discuss about the structure of this microsporangium. So this microsporangium is generally surrounded by four layers of wall: epidermis, endothecium, middle layer and tapetum. So outermost blue layer is epidermis, second layer is called endothecium, Third two layer is called middle layer, and fourth this pinkish color layer I have shown you is tapetum. And below the tapetum there are the microspore mother cell. Mother cell means microspore mother cells means microspores are produced from this cell. That's why they are called microspore mother cell. The outer three layer protective and help in dehiscence. So outer this three layer function is the help in dehiscence means spreading of the anther only. And anther release to the pollen grain. The innermost layer means tapetum is the innermost layer out of the four layer. Tapetum is the innermost layer which provide nourishment to the developing pollen grain. And innermost cells, these purple color cells are known as sporogenous tissue. It is compactly arranged homogeneous cells which are present at the center of each microsporangium when anther is young. From each cell, microsporangium is produced. Next, microsporogenesis. Microsporogenesis means the process of formation of microspore from PMC means pollen mother cell or microspore mother cell. Same thing. Student, please don't confuse. Pollen mother cell or microspore mother cell is the same thing. So, the process of formation of micro means microspore and microspore genesis means formation of microspore is called microsporogenesis. You just remember like that, then it will be very easy for you. Next, each cell of the sporogenous tissue has the capacity to give rise to a tetrad. Hence, each cell is potential pollen or pollen mother cell. Means from the sporogenous tissue, look, from this sporogenous tissue, purple color cell, each cell give rise to four pollen mother cell. From one sporogenous tissue, four cells are produced. As anther mature, the microspore get detached from each other and develop to pollen grain, means male gametophyte. Now come for the diagram. So this is the dissection of the here four microsporangium are produced within the microsporangium. These are the microspore mother cell. So from microspore mother cell, look, from each microspore mother cell, four pollen haploid microspore are produced, and that's why it is called microspore tetrad. Tetrad means four, and from each microspore one pollen grain are formed, and within the pollen grain there are two cells. One is called generative cell, one is called vegetative cell. So from the sporogenous tissue, from each cell of sporogenous tissue, microspore mother cell is produced. From each microspore mother cell, four haploid microspore are formed. From each microspore one pollen grain is formed. Next, structure of pollen grain. So pollen grain has two layers, exine and intine. Exine is the outermost layer, which is hard, made up of sporulinin, and another is intine is the innermost layer, which is continuously made up of cellulose and pectin. So student, I am giving the notes point-wise. If you go through this point, no need to go through your NCERT book. Every important point I have touched here. So next, vegetative cell and generative cell. In this diagram, look, I have already shown you within the mature pollen grain, there are two cells. One is called generative cell, another is vegetative cell. So now we are going to discuss about generative and vegetative cell. Vegetative cell is the bigger one. First, it is a bigger one. It is abundant food reserve, means food are reserved in these cells. Large irregular nucleus is present and responsible for the development of pollen grain. It means it provides food to the developing pollen grain. Generative cell is small, involved in syngamy, means fusion of syngamy means fusion of gametes. So in fertilization it takes place, and then cytoplasm and nucleus is present. Now go for the structure of a mature pollen grain. So first you go through this pollen flow chart. So pollen mother cell, from pollen mother cell, pollen tetrad means four microspore is produced. That is, it is called pollen tetrad. From pollen tetrad, pollen grain are formed. Within the pollen grain, vegetative cell and generative cells are produced. So pollen mother cell undergo meiosis one division, divided into two and then divided into four. And this four cell stage is called pollen tetrad. And from pollen tetrad, look, four pollen grains are formed. Next, this is the structure of a mature pollen grain. This is the outer covering, that is called axine, and this is the is called intine. There are two cell. One is called generative cell or generative nucleus, and another is called vegetative cell or vegetative nucleus or tube nucleus. So don't confuse vegetative nucleus or tube nucleus is the same thing. And intine is thin. This inner layer is called intine. Outer layer is called axine. Next, pollen grain. And in 60% angiosperm, pollen grain are set at two cell stages, while the other generative cell undergo meiosis to form two cell male gametes. It means at this stage, the pollen grain is ready for pollination. Means at this stage, with the help of pollination, it go and sit on the stigma and they stick to the stigma where next steps for fertilization takes place. Next, go difference between microspore and pollen grain. So microspore is the small haploid structure that give rise to male gamete, and pollen grain is the small haploid structure that gives rise to the male gamete. Microspore gives rise to the male gametophyte, whereas pollen grain gives rise to the male gamete. Found in both seedless and seed plant, found only in seeded plant. Here I am showing you five or six difference. You just remember two, three, it will be more than sufficient, because sometimes difference come and this type of question will carry 2 marks. Next, gynosium and formation of female gametophyte. The gynosium represent the female reproductive part of flower, and in some it may be monocarpillary, means one pistil is present. If too many is present, then it is called multicarpillary, and it may be syncarpous or apocarpous. So syncarpous, look, so when the pistils are jointed at the tip, that is called syncarpous. If they are free from each other at the tip, that is called free carpel or apocarpus. Next, this is the structure of a carpel. It has three parts: stigma, style, and ovary. Within the ovary, the ovule is present. Now go for the megasporangium or ovule, structure of megasporangium. The ovule is attached to the placenta by funicle, means the stalk of the ovule is called funicle. And the ovule, in the center, there is a portion that is called nucellus. And the point where this point, look, stalk, and this nucellus is covered by two covering. These two covering called integuments. These two are integuments. This is inner integument. This is outer integument. And the stalk, means funicle, attached at the portion of the integument. That joining point is known as hilum, and within the nucleus there the embryo sac is present. So this is the structure of a megasporangium or ovule. Or if question come, draw the structure of a mature female gametophyte, then also you have to draw the diagram of this diagram. Next, megasporogenesis, means how mega means female. Female spores are produced. That is called megasporogenesis. The megaspore mother cell, means MMC. In case of male, we tell it at PMC, means pollen mother cell. Here it is called MMC, megaspore mother cell, get converted into megaspore by the process of megasporogenesis. The MMC is large and contain a dense cytoplasm and a prominent nucleus. It undergo meiosis to produce 4 megaspore. So look, this is the structure of a mature ovule. Here within the nucellus, lots of cells are there, but the cell which is just at the point of the micropylar end present, that cell enlarges and it is converted to MMC, means microspore mother cell. Look, this converted to microspore mother cell. Then this microspore mother cell undergo division and finally it will form 4 number of cells. Look, these are the megaspore tetrad. So out of this tetrad, what happened? This first 3 cell degenerates, means they will be destroyed and only the lowermost cell will remain active and that will convert it into a megaspore. It means from one megaspore mother cell, only one megaspore is produced. Whenever in case of male, from one pollen mother cell, 4 pollen grain are formed, but in case of female, from one megaspore mother cell, only one megaspore is produced. Next, female gametophyte. The most flowering plant, only one megaspore is functional when other three are degenerate. Already I have explained. So the single functional megaspore develop into female gametophyte, and this type of development is called monosporic development. Because monosporic means from single cell it is developed into female gametophyte, that is called monosporic development. The nucellus of the functional megaspore divide mitotically to form two nuclei which move towards the opposite end, forming two nucleate embryo sac. And finally, 4 nucleate and 8 nucleate stages. Look, I have shown you in the diagram, then you will be easily understand. So look, this is the megaspore mother cell. So it first form into 4 megaspore tetrad. From 4 megaspore tetrad, the upper 3 will be degenerate and only the lowermost become active. And this is converted into megaspore. Then the nucleus is divided into 2. Again they divided and finally divided into 4. Again they divided and finally divided into 8. And when they become 8, nucleus number become 8. Out of 8, 3 will go to this side and 3 will go to this side and 2 come into the center. So this 3 cell will call antipodal cell and this 3 cell will call egg apparatus. Egg apparatus means in between from these 3 cell the center one is called egg cell. Look, here is labeling, center one is egg cell, other 2 cell are called 2 synergids. And the in the center two nucleus, two cell come and they are jointed and become a diploid cell which is known as polar nuclei. So this is the cell division of a female gametophyte. So look, here it is sitting. The 3 of the 6 cells are placed at the micropylar end and constitute the egg apparatus. Egg apparatus mean question may come. Then you have right egg apparatus mean 2 synergies plus 1 egg cell. And out of this, I will show you in this diagram. Wait. So in this diagram, look, in this diagram, so these 3 are called egg apparatus. These 2 are synergies. In the centermost, it is called egg cell. In the center, it is called polar nuclei. And these 3 cell are called antipodal cell. And this direction where these integuments are have a little opening, that side is called micropylar end. So the 3 cell which are towards the micropylar end are called egg apparatus, and the opposite side of the micropylar end is called callosal end, and the these three cell which are situated towards the callosal end is known as antipodal cell. So this is callosal pole. 3 cell towards the callosal pole is called antipodal cell. The 3 cell towards the micropylar end is called egg apparatus, and the center 2 cell is known as polar nuclei. So student, today we have discussed about the structure of male gametophyte, structure of female gametophyte, how the pollen grain are produced, how the female organs, female gametophyte are produced. So we have discussed. In our next video, I am going to discuss about the pollination and double fertilization. So till then, okay, goodbye.